Drugs. How the drugs are specifically used by the body to the specific site of illness or injury. Drugs are designed to target specific sites in the body to treat illness or injury. The way a drug is used by the body depends on the drug's chemical properties and the route of administration. One of the most common routes of administration is oral, which means the drug is taken by mouth. Oral drugs are usually in the form of tablets or capsules, and they are absorbed into the bloodstream through the digestive system. Once in the bloodstream, the drug is transported to its target site, where it can have its intended effect. Another common route of administration is intravenous, where the drug is injected directly into a vein, typically through a needle and syringe. This route is often used for drugs that need to act quickly, such as during emergency situations or for drugs that are not well absorbed when taken orally. Topical administration refers to drugs that are applied directly to the skin, mucous membranes, or other external surfaces of the body. These drugs are usually in the form of creams, ointments, or gels, and they are designed to target specific areas of the body, such as the skin or eyes. Other routes of administration include inhalation, sublingual, rectal, and intramuscular. The choice of route of administration depends on the drug's properties and the condition being treated. Once the drug reaches its target site, it can interact with specific receptors or enzymes to produce its desired effect, such as killing bacteria, reducing inflammation, or relieving pain. However, drugs also can produce unwanted side effects in other parts of the body, depending on the drug's properties and dose. Pharmacokinetics of a drug. Pharmacokinetics is the study of how a drug moves through the body, including how it is absorbed, distributed, metabolized, and eliminated. Absorption. This is the process by which a drug enters the bloodstream. The rate and extent of absorption can be affected by a number of factors, including the route of administration, example, oral versus intravenous. The formulation of the drug, example, tablet versus solution, and the presence of food in the stomach. Distribution. Once a drug enters the bloodstream, it is distributed to various parts of the body, including the brain and organs. The distribution of a drug is determined by its ability to cross cell membranes, as well as by the binding of the drug to plasma proteins. Metabolism. Many drugs are metabolized, or broken down, by the liver and other organs before they are eliminated from the body. The metabolism of a drug can affect its efficacy and safety. Elimination. The final step in the pharmacokinetic process is the elimination of the drug from the body. This can occur through various routes, including the kidneys, in the form of urine, and the liver, in the form of bile. Understanding the pharmacokinetics of a drug can help to optimize its therapeutic effect and reduce the risk of adverse reactions. Pharmacodynamics of a drug. Pharmacodynamics is the study of how a drug interacts with the body to produce its effects. It is the mechanism of action of the drug, including the specific receptors, enzymes, or other proteins with which the drug interacts, and the downstream effects that result. Receptor binding. Many drugs bind to specific receptors in the body, which are proteins found on the surface of cells. This binding can activate or inhibit the activity of the receptor, leading to changes in cellular signaling and ultimately resulting in the drug's effects. Enzyme inhibition/induction. Some drugs interact with enzymes, which are proteins that catalyze chemical reactions in the body. A drug can inhibit an enzyme, reducing its activity and slowing down a particular metabolic pathway. Alternatively, a drug can induce an enzyme, increasing its activity and speeding up a metabolic pathway. Pharmacokinetics-pharmacodynamics relationship. The relationship between the pharmacokinetics and pharmacodynamics of a drug is critical in determining the drug's therapeutic effect. The concentration of a drug at the site of action and the duration of time it remains at that site will determine the effect of the drug. Agonist and antagonist. Drugs can be classified as agonists or antagonists depending on their interactions with receptors. Agonists bind to receptors and activate them, leading to a response similar to that of the natural ligand. Antagonists bind to receptors but do not activate them, thus blocking the action of agonists and the natural ligand. Pharmacodynamic modeling. Mathematical models are used to study the relationship between the drug concentration and its effect, which are known as pharmacokinetic pharmacodynamic, Part PD, Models. These models can help to understand the dose-response relationship of a drug, which is important for drug development and optimization of therapy. Understanding the pharmacodynamics of a drug can help to optimize its therapeutic effect and reduce the risk of adverse reactions. It also helps in the selection of the appropriate drug for a particular patient and in the development of new drugs. What role liver plays in the metabolism of the drugs? The liver plays a crucial role in the metabolism of drugs in the body. The liver is responsible for metabolizing, or breaking down, the majority of drugs that are taken orally. The liver's primary role in drug metabolism is to make the drugs more water-soluble, so that they can be excreted from the body more easily through the urine or bile. The liver is home to a diverse array of enzymes that are responsible for metabolizing drugs. These enzymes are found in the liver cells called hepatocytes. These enzymes include cytochrome P450 enzymes, which are responsible for breaking down a wide variety of drugs. The cytochrome P450 enzymes are responsible for breaking down about 75% of drugs that are metabolized by the liver. The liver also plays a role in regulating the amount of drugs in the bloodstream by controlling the rate at which drugs are metabolized. When drugs enter the liver, they are either metabolized and eliminated from the body or they are stored in the liver until they are needed. Drug metabolism. Drugs that are metabolized in the liver can have interactions with other drugs. Some drugs can inhibit the activity of certain enzymes, which can slow down the metabolism of other drugs, leading to higher levels of the drug in the bloodstream and an increased risk of side effects. Similarly, other drugs can induce the activity of certain enzymes, which can speed up the metabolism of other drugs, leading to lower levels of the drug in the bloodstream and reduced effectiveness. It's important to note that liver disease can affect the metabolism of drugs and change the way a person responds to a medication. People with liver disease may need to take lower doses of a medication or take different medications, which would be decided by the healthcare professional. Half-life of a drug. Half-life refers to the amount of time it takes for half of a drug to be eliminated from the body. This can vary depending on the drug and the individual taking it. The half-life of a drug is important because it determines how frequently the drug needs to be taken in order to maintain a consistent level in the body. Dosage of a drug. Dosage is the amount of a drug that is prescribed to be taken at a particular time. The dosage of a drug is determined by several factors, including the individual's age, weight, and overall health, as well as the condition being treated and the specific drug being used. When determining the dosage of a drug, doctors consider several factors, including 1. The half-life of the drug. The longer the half-life of a drug, the less frequently it needs to be taken. For example, a drug with a short half-life may need to be taken multiple times a day, while a drug with a long half-life may only need to be taken once a day. 2. The therapeutic range. This is the range of drug concentrations in the body that are known to be effective in treating a particular condition. The goal is to keep the drug concentration within this range, while minimizing the risk of side effects. 3. The patient's response to the drug. The patient's response to the drug is also taken into account when determining the dosage. If the patient is experiencing side effects or the drug is not providing the desired therapeutic effect, the dosage may be adjusted. 4. The patient's renal and hepatic function. The patient's renal and hepatic function will be considered as these organs play an important role in metabolism and excretion of drugs. Dosage adjustments. Once the dosage of a drug has been determined, it is important to closely monitor the patient's response to the drug and make adjustments as needed. This may involve adjusting the dosage or switching to a different drug if the current one is not providing the desired therapeutic effect. It is important to note that the dosage of a drug should always be determined by a qualified healthcare professional and should not be adjusted without their guidance. Drug resistance. Drug resistance refers to the ability of bacteria or viruses to resist the effects of certain drugs that were previously effective in treating them. This can occur when the bacteria or viruses develop genetic mutations that allow them to survive exposure to the drugs. As a result, these drug-resistant strains can continue to multiply and spread, making them more difficult to treat. Bacteria can develop drug resistance in several ways, including natural selection. Bacteria that are exposed to drugs will typically die, but a small number may survive due to genetic mutations that make them resistant to the drug. These resistant bacteria can then multiply and spread, leading to a population of drug-resistant bacteria. Horizontal gene transfer. Bacteria can also acquire resistance genes from other bacteria through a process called horizontal gene transfer. This can occur through the transfer of plasmids, small, circular pieces of DNA, or through the exchange of genetic material through bacterial conjugation. Overuse of antibiotics. The overuse and misuse of antibiotics can also contribute to the development of drug-resistant bacteria. When antibiotics are used excessively or inappropriately, they can kill off susceptible bacteria, leaving behind only the resistant strains to multiply and spread. Viruses can also develop drug resistance, although the mechanisms are different from bacteria. For example, viruses can develop mutations in their genetic material, such as their RNA or DNA, that can make them resistant to antiviral drugs. Some viruses, such as HIV, can also become resistant to drugs by developing multiple copies of the enzymes that the drugs target, which makes it more difficult for the drugs to be effective. In addition to the natural evolution of resistance, in some cases, bacteria and viruses can develop resistance to drugs due to the selective pressure of the drugs in the environment. For example, overuse of antibiotics in agriculture can lead to the development of antibiotic-resistant bacteria in animals, which can then be transmitted to humans. To combat drug resistance, it's important to use drugs appropriately, to avoid overuse and misuse of antibiotics, and to invest in research and development of new drugs. Additionally, proper infection control measures, such as hand hygiene and isolation of infected patients, can help to slow the spread of drug-resistant bacteria and viruses. Cross-resistance of a drug. Cross-resistance refers to the phenomenon in which microorganisms that have developed resistance to one drug also have resistance to structurally or functionally related drugs. This is because these drugs target the same or similar cellular pathways. For example, bacteria that have developed resistance to a beta-lactam antibiotic, such as penicillin, may also be resistant to other beta-lactam antibiotics, such as amoxicillin or cefotaxime, because they all target the same bacterial enzyme, called penicillin-binding proteins. Similarly, bacteria that have developed resistance to a certain class of antibiotics, such as the tetracyclines, may also be resistant to other drugs in the same class, such as doxycycline or minocycline, because they all target the same bacterial protein, called the 30S ribosomal subunit. Bacteria and viruses can develop resistance to drugs through several mechanisms, including mutation and horizontal gene transfer. When a microorganism requires a genetic mutation that confers resistance to a specific drug, it may also be resistant to similar drugs because they target the same or similar cellular pathways. This is known as cross-resistance. Additionally, bacteria can transfer genetic material, including resistance genes, to other bacteria through horizontal gene transfer mechanisms, such as plasmids, making them resistant to multiple drugs. Ingredients of a drug. Most drugs contain chemicals as their active ingredients, but some drugs, such as herbal supplements, may contain natural substances as well. Additionally, some drugs may contain a combination of chemicals and natural substances. Many drugs are made synthetically. Synthetic drugs are created using chemical processes and are often designed to mimic the effects of natural compounds. They are typically made in a laboratory using a variety of chemical compounds and are usually manufactured on a large scale for commercial use. Examples of synthetic drugs include many types of painkillers, antidepressants, and anti-anxiety medications. Expiry of drugs. The U.S. Food and Drug Administration, FDA, is responsible for determining expiration dates for drugs. The FDA sets expiration dates based on stability testing, which is conducted to determine How long a drug remains potent and safe when stored under specific conditions, such as temperature and humidity. The expiration date is the date after which a drug may no longer be at its full potency and may not work as well as it should. When a drug is first approved by the FDA, the manufacturer must provide stability data that demonstrates the drug's potency and safety over time. Expiry. The FDA then uses this data to establish the expiration date for the drug. The expiration date is based on the time period during which the drug is expected to remain potent and safe when stored under the conditions specified on the label. Once the drug is on the market, manufacturers are required to continue testing the drug's stability and to update the expiration date as necessary. Additionally, FDA also performs post-marketing surveillance for drugs to ensure their safety and efficacy. It's important to note that expired drugs may still be safe, but the potency may have decreased over time. So the FDA advises to use them before the expiration date to ensure that you are getting the full benefit of the medication. Drugs can lose their potency over time, especially if they are not stored properly or if they have passed their expiration date. The expiration date is the date after which a drug may no longer be at its full potency and may not work as well as it should. However, it does not mean that the drug becomes completely ineffective or harmful after the expiration date. The potency of a drug can degrade over time due to various factors such as changes in temperature, humidity, and exposure to light, which can cause chemical reactions that can alter the drug's composition. It's important to note that some drugs can be effective even after their expiration date, and some may lose potency well before their expiration date. So, expiration dates are often conservative estimates. In case of emergency, expired drugs can still be used with caution, but it's best to check with a healthcare professional for guidance. In general, it's a good idea to store medications in their original containers in a cool, dry place, away from light, and to use them before their expiration date.